Okay, this is going to be review for Unit 4, and we're doing it in 2 parts. So the part A is going to include the cell communication. So we'll go over signal transduction, change in transduction pathways, about reception as well. And then that comes back, cell communication, when we do gene expression. So you want to keep that in mind, that those units are linked, Unit 4 and 6. And then we're going to look at feedback and then cell cycle and regulation cell cycle in part B. So you have your explaining biological concepts or processes as well as describing them. You're having justifying a claim by connecting it to evidence and predicting causes and effects to when you change a component of a system. And for cell communication, we modeled cell communication, and we also did our POGL on cell communication as well. So we looked at 3 types of cell communication. We looked at direct contact, and the example that we used there was the immune system. We looked at short distances. The example that we used for short distances was a little bit in the immune system, but we also looked at short distances with synaptic signals in the nervous system, and then long distances, which we looked at with the endocrine system. So direct contact, these are cells communicating with cell-to-cell contact. So example in organisms that we're going to use is the human immune system, in animals, and then plasmodesmata in plant cells. So in the immune system, if you remember, the macrophages or your other dendritic cells ingest a bacteria or a virus, another antigen being anything that causes immune response. And when it engulfs it, it takes a little piece of it and it puts it on its major histocompatibility complex 2. This is the one that is shared by all immune cells, right? And now we refer to it as an antigen-presenting cell. It's going to release a chemical signal. The general name is cytokines. This specific one is called interleukin, inter between leuc for white, right? Because they're going to involve the white blood cells. And then that interleukin brings the T helper, which is going to bind to your major histocompatibility complex 2. It uses a protein called CD4 to do that. And then it releases its own interleukin 2, and that's going to activate other cytokines. It's also going to cause more T helpers to be activated as well. Okay, so here's our example, and this is in your book, or if you go back and you look at your notes from the immune system, you'll see this, or cell signaling. So you have the bacteria eaten, takes a little piece of it, it puts on the MHC2. Here's that CD4. Okay, so that's helping bind the MHC2 to the T cell receptor. So direct contact here, right? Here you see that interleukin 1, right? So there's that cell signal. That's going to help it differentiate, and it's going to release the interleukin 2, which is going to help bring more T helpers. And that's going to be important for your cell-mediated response, which is going to involve your cytotoxic T cells, and your humoral response, which is going to involve your B cells. Okay. Plasmodesmata, those are like gap junctions that we have in animal cells. Those are openings here in the cytoplasm. It allows things like transcription factors and regulatory RNases to pass between neighboring cells. Plants can then coordinate how their tissues or organs respond to environmental stress, or if they're growing or they're developing. So they become very important for that reason. All right. So we looked at the short distances. You can have local regulators, all right, and they're going to use diffusion. You can have autocrine, that's a signal that it's created by the cell itself, like the T helper, and it acts on itself. Auto, remember, always means self. Paracrine, that's a nearby one, and the example we used was in the nervous system when we talked about synaptic signaling, all right? But some other ones that you should be aware of, the plant immune response, form sensing, and then morphogens and embryonic development. This is going to tie again back the morphogens embryonic development to Unit 6. So quorum sensing, there's a really good TED Talk by Bonnie Bassaron. She was the one, the scientist who discovered it, and I think it's 18 minutes. And if you just put in TED Talk quorum sensing, it will come up. So, and students usually enjoy that particular TED Talk. She's very enthusiastic. So it's the process of cell-to-cell communication, bacteria cells. So the bacteria cells, just to simplify it, they actually are going to release signals that give information to each other about the density, right? Some of these will work within a species, some of them will be between species, but it allows them to change their gene expression. And so they can coordinate together. For instance, if you have an infection, they're actually going to ensure that there's enough of them before they start to attack your cells, right? So that they won't all be killed off by your immune system right away. So it's allowing them to coordinate. They'll also use it if they're too dense. Let's say if they're not pathogenic, to stop reproducing because there's not enough resources. So it's a way for them to communicate together and work together, right? And this is the Dr. Bassler's TED Talk. Really do recommend it. I mean, it's worth seeing how they behave. Neurotransmitters, these are local regulators. So if you remember, we went over how the nervous system worked, but I'm just going to focus at this point on the cell signaling piece of it. So at the end of your axon, that electrical chemical signal that changing from a negative 70 millivolts to at least hitting the threshold potential of negative 55 millivolts, that's going to cause these calcium voltage gated channels to open. Calcium comes in, that causes the exocytosis of the synaptic vesicles. Those contain your neurotransmitters. Those diffuse across the synapse. They're going to bind to receptors on the postsynaptic cell. And that's going to cause gated ion channels to open, either sodium gated ion channels if it's excitatory, or if you want to stop the impulse, it's inhibitory, it'd be a potassium gated ion channel. Okay. And the reason why the sodium is going to be excitatory is because if we see here, you go from negative 70 millivolts to the threshold, which is negative 55 millivolts. And then we're going to have the signal cascade down, the action potential cascade down the axon. If it's inhibitory, you know, it's going to go, if there's potassium, the potassium is going to flood out. So you're going to lose lots of potassium, and it's going to go below negative 70 millivolts, and that's going to be inhibitory. So that's when you want to stop a response. You're getting information that's not useful for you. Okay. Afterwards, a couple of things can happen to the neurotransmitters. They can be degraded, okay, or they can be reabsorbed. Right. So that's called reuptake. This is how some SSRI will work. That's one of trying to think, like, I think Zoloft is an SSRI that's used to treat things like depression. So keep something like serotonin in the synapse a little bit longer so that it can hit these receptors and send the signal. In the plant immune system, salicylic acid, which you may have seen on some products, usually used for skin and stuff like that, that's produced by plants in response to a pathogen. So it's going to activate genes involved in defense. The stomata or stoma are going to close, right? Kind of like you're closing off the system. You're going to start to make some antimicrobial proteins, and then it's going to do some targeting and destroying of non-plant nucleic acids. So you can see it's really important in this immune response here. And then morphogens, we talked about this with embryology a bit. These are going to give positional information. So Spearman's experiment on the primary organizer, when it starts gastrulation in the embryo during implantation, the primary organizer is right here on the blastopore lip, and it sends out a chemical signal. And so what you end up having is the neural tube form above here. If, in his experiment, he took from a gastrula, he put a second blastula on another gastrula. And so he created conjoined twins that way. Our long-distance signals that travel by the circulatory system to our target cells is in our endocrine system. So the target cells have to have a specific receptor for the signal. And we looked at a few types of signals. We looked at hydrophobic signals. They have the receptor inside. It can either be in the cytoplasm or it can be in the nucleus. And when that hydrophobic signal binds to the receptor, it's going to act as a transcription factor. So it's going to cause genes to be expressed. And again, this loops right back into Unit 6. Hydrophilic signals, the receptor will be embedded into the plasma membrane. So if you remember, we did that activity, and that was one of the issues we had with the activity that we were testing out. Right? So here you can see the hydrophilic. Here's the hydrophobic. Our hydrophobic signals are going to include testosterone, estrogen, progesterone. It's also going to include our glucocorticoids and our adrenal steroids as well. Okay, so our long-term stress response. So hydrophobic, those are steroids. Remember, that's a lipid-based 4-carbon rings, and they're going to bind to that internal signal, and they're acting as a transcription factor. Right? And this is a really great picture off here on the right side that does a good explanation of that. Estrogen would be a good example. That's made in the ovaries. It has multiple effects beyond your sexual secondary characteristics. All right, it adjusts temperature, memory. It's going to be reducing cholesterol in the blood, protects the heart from cholesterol. So multiple effects, has different effects in different parts of the body, right? Because even though this estrogen's coming in, this receptor could be different. And therefore, it's going to act as a transcription factor on a different gene. Testosterone will work. It is a steroid, again, based hormone. It's made in the male testes. There's a small amount made in the female ovaries and a small amount made in the adrenal glands, right? And again, we see things like red blood cell production, maintenance of bone mass. Those are all things that are controlled by testosterone. Right? So and that's going to be generating, transmitting, receiving, and responding to the signal. So we're going to look at the hydrophilic one, the reception here, which hydrophobic has as well. But then hydrophilics have the signal transduction pathway, and then both of them have a response. We've already talked about the hydrophobic, so I'm going to focus now on the hydrophilic signals, the protein-based ones. Okay, this is an example FRQ from 2013. Okay. So you would have to look at this and explain each of the roles of the numbered steps regulating gene expression. So you would say, step 1, I see a reception of a hydrophilic signal. And I know that's it because the receptor is in the plasma membrane. Step 2 is my signal transduction pathway, which is amplifying my signal. And step 3, I have the response, a transcription has been made that's changing the gene expression. And you can look this up. It's released, so you can look it up and check. We have our receptor transduction, right? And so that's a number of different ranging a bunch of different things. So again, hydrophilic peptide hormones recognized by a specific receptor on the plasma membrane. We went through 3 different types of receptors. We went through the G protein receptor. We went through the kinase receptor, did the GATA receptor. At that, specific at this, I'll talk a bit about the protein receptor. Again, by receptor, and then the cell, the part of that inside the cell is going to change transduction. So we have insulin as an example, hormones, and epinephrine, illustrative examples. We went through all of these. So insulin, if you remember, that's related to diabetes. If you don't make insulin, you have type 1 diabetes. Receptors become desensitized to insulin. That's type 2 diabetes. So insulin itself is released from the pancreas, the beta cells. It's a tyrosine kinase receptor, so it has the two receptors. They each bind insulin, then they come together. And then they have 6 different kinases, so they can start multiple pathways that could do different things. And insulin's job is to lower the blood glucose, and it does that by having your liver or skeletal muscles uptake blood glucose and turn it into glycogen, or your adipose tissues store the excess glucose as lipid or fat. Human growth hormone, that is made in your anterior pituitary gland. It actually receives a message from the hypothalamus. Growth hormone releasing hormone, and that is going to act on that anterior pituitary gland to release human growth hormone. So it causes muscle growth, it's going to cause bone growth. And then your thyroid one, this is important, this controls your metabolism or bioenergetics. So it helps your heart rate, your blood pressure, your muscle tone. And so your hypothalamus is going to release TRH, and then that acts on the anterior pituitary, that releases TSH, and then that acts on the thyroid, which is right here, and that's going to release T3 and T4. T3 and T4 are going to help control your metabolism. You can see here negative feedback, so high T3, T4, you're going to turn off, I hate that my computer does that, your anterior pituitary, and then that can also turn off the hypothalamus, and TSH can turn off the hypothalamus. And then epinephrine, this was a G protein receptor. So epinephrine is produced by your adrenal glands that sit on top of your kidneys, and it's a short-term stress response. You release epinephrine or adrenaline, right? And that binds to a G protein receptor, which then is going to work on the G protein, which is interior, so the green thing here, and that's a peripheral protein. So the G protein is going to become activated by the GDP coming off, GTP going on. And that's going to activate actually adenylene cyclase. Adenylene cyclase is going to make CAMP, and that'll be a secondary messenger that's going to activate a protein kinase cascade. And so, for instance, your liver, you're going to break down that glycogen, release glucose, so you have enough energy for your fight or flight response. So, our gated ion, our ligand panels, those are here. The example that they're showing here is the neurotransmitters, opening those up. We also have the G protein. That's going to, it's not on this, oh no, I do have a G protein here. That is going to be found with the epinephrine and release of glucose from glycogen. Okay, so you can see here as, let's say, the ligand binds. Ligand is just a general term for something that binds to receptor. Then my G protein coupled receptor, that G protein, that's GDP taken off in GTP, and now it's going to activate another enzyme, and I can have a second messenger activated. All right, a little bit better here. Signal transduction, that's going to link the reception to the response. So we're going to see that we're going to have these protein kinase cascades. They're going to be deactivated by phosphatases. That's not shown in this picture. Now, it amplifies the inner process, and it allows it to react differently to the same chemical messenger, because you can have different cascades happening. Okay, and this is your epinephrine pathway again. That was the example that we used quite a bit. Second messengers, they relay the signals by the receptor inside the cell. All right, so the signal is picked up by the receptor, right? And then inside here, inside the cell, or sometimes inside the nucleus, it's going to cause that change, right? So again, it's going to amplify the signal, and it can trigger phosphorylation cascades. Two examples that we looked at was CAMP and calcium. And here you can see the calcium is being released from the endoplasmic reticulum. And that's going to activate, in this case, a bunch of proteins, and you're going to have multiple cellular responses from a single messenger. So it really allows amplification and for different responses to occur within the cell. So CAMP That's actually ATP that's lost to phosphates, so it's a monophosphate, and the C is just because it gets that cycle. It looks like a circle, okay? And that's going to do a signal transduction pathway. And here you can see this is the epinephrine when we get more than one response. We get an inhibition of making glycogen, and then we get the promotion of glycogen being broken down. So we've activated proteins and enzymes to do both. Okay, so cell responses can, again, they can be a change in gene expression or a cell function, or it can even be apoptosis, okay? So steroid hormone response, that's usually going to be turning on or off our gene when it binds to its receptors, acting as a transcription factor. And a good example is testosterone. It triggers the expression of the SRY gene for maleness. And so we talked about XY females. Quite often they don't have this SRY gene. So when you start off as a female, and then you get that testosterone burst, and if you have that SRY gene, your development will go off as a male. If they are lacking that SRY gene, even if they have a Y chromosome, they will continue on as a female. And then some cells are going to respond to gases, and I'm just using ethylene as the example because that's the illustrative example in the CDD. But we have examples for humans as well of gas hormones. So here, ethylene causes a change in enzymes, and because it's going to activate all these enzymes, hydrolase, pectinase, kinase, amylase, those all are going to cause the fruit to ripen. Right? So amylase breaks down starch to sugar. Pectinase is going to cause the fruit to get soft. Hydrolase is going to cause the fruit to get green to red color so that organisms would know. All right. So they're turning on or off. It can cause cytosis. The internal environments, pH, apoptosis, and what you should know are insulin, cytokines, and then the upper half. And I will give you a list of illustrative examples as well. I'll post that on Classroom. Mutations in the receptor proteins, that's going to alter the cell's response. So an example is the mutation in the HER2 receptor. You may have seen that in commercial. That can cause the cells to grow ugly. That's a cause of some forms of breast cancer. Another one you may have seen again in commercials for different medicines, you hear about psoriasis. So this is a picture of psoriasis here. And it's a mutation in the epidermal growth factor. And so it causes these scaly plaques and patches to occur on skin. And it's supposed to be quite uncomfortable to have this. Okay, so chemicals that interfere with the signal pathway, those can actually either activate or inhibit a pathway. A good example are pesticides. So a number of pesticides are going to be either working to block the breakdown of acetylcholine, or they're going to cause the chitin synthesis pathway so that the pest isn't making its exoskeleton. By blocking or stopping the acetylcholine from being picked up, it overstimulates your nerve cells, and that causes the death of the pest or the insect. Some pesticides that were used quite a while ago, they actually affected human neural pathways as well. So that's why, we try to decrease our use of pesticides, as well as microevolution. Then you get pesticide-resistant insects. Another example, antihistamines. If you have hay fever or allergies, you get that inflammatory response, and that's because your eosinophils and your immune system release histamine, and that causes swelling, red eyes, runny nose, your eyes are itchy and they're watering because your body is misinterpreting what that normal pollen in the environment is, and it's treating it as though it's a parasitic worm, right? So we want to block that because it's not a useful response, and so that's what antihistamines do, like Benadryl. So they are going to block the histamine receptor, and so you can't have that inflammatory response start to occur. All right, and this is another example. This is released, so it's publicly released. You can look at it, and this is from 2010. Right, so it shows you how you could be asked about this. Again, I would see this being put in with a science practice. I'm making this before we know on April 3rd. So once we know on April 3rd, we'll have a better idea of the types of assays. Okay, and that is all for part A.